Good morning, everyone. Take off my mask. So good morning. Thanks for being here, here in Glasgow or remotely from home. So it's a great pleasure this morning to have this keynote lecture from Dr. Zalesky. My name is Anne Gallagher. I'm from University of Montreal, Canada. I'm co-chairing this session with Nicola Palomero Gallagher from Research Center ULIC. And a few words on Dr. Zalesky before he starts. So he's an associate professor in engineering and medicine at the University of Melbourne, Australia. He completed his PhD in electrical engineer at the same institution in 2007. Adapting an engineer mindset to study the brain has enabled him to innovate novel methods and models to understand the network organization of the human connectome in health and disease. He established one of the most widely used tools for performing statistical inference on connectomes, co-authored the first textbooks on brain network analysis, and led development of the Melbourne Subcortex Atlas. Dr. Zalesky is ranked among the top 1% of researchers worldwide, according to citations to his work, and he has recently voted by his peer as one of Australia's most innovative biomedical engineer. So it's a real honor having him here this morning. I won't take more of his time, so Dr. Zalesky.
Thank you very much for the introduction. So I'd like to begin by thanking the organisers for giving me the opportunity to present some of our group's research. I'm going to present three separate projects that we're currently undertaking. The first one is a new area of research, and that's in understanding brain-body interactions in aging and disease. I'll then move on to some of our translational work, our clinical translational work, where we're harnessing individual brain variability to optimize brain-based therapeutics. And finally, I'll wrap up with some of our more traditional work in developing methods for mapping and modeling human connectomes. So we're all here at OHBM in Glasgow because we're interested in studying and understanding human brain structure and function. However, as neuroimagers and neuroscientists, we often forget or overlook that the brain is densely interconnected with virtually every other organ and body system. And this is not only via the 50 or 60 kilometres of axonal cabling comprising the peripheral nervous system, but also through metabolic, hormonal, and immunological signalling pathways. Moreover, more recently, there has been growing interest in particular brain-body axes, such as the brain-heart axis, the brain-gut axis, and their importance is recognised as critical to optimal brain and body health across the lifespan. So in the first part of my talk, I want to present some of our preliminary work in understanding brain-body interactions, particularly through the lens of aging. And I hope that I can convince you that as neuroimagers and neuroscientists, if we are to truly adopt a systems approach to studying brain structure and function, we should broaden our horizons and embrace the importance of understanding how the brain is influenced by and influences other organ and body systems. So first I want to begin with the concept of brain age. I mean, there have already been a lot of workshops and symposia at this year's OHBM on this concept, and there's a burgeoning literature on estimating age and its associations with various lifestyle and environmental factors. And as I've shown there on the right, for example, things such as musical talent is associated with a younger appearing brain. And in the top right there, we see that neurodegenerative disease of the brain and body are associated with older appearing apparent brain age. What is the brain age concept? For those of you that are not familiar with the concept, the core idea here is that we take any of a number of neuroimaging phenotypes and we train a model to predict an individual's chronological age. And this can be anything from a simple regression all the way up to a convolutional neural network. The age gap is then simply defined for an individual as the predicted age minus the chronological age. And then we see if the gap is greater than 0, this suggests that an individual's brain appears older than same-aged peers. Conversely, if the gap is less than 0, the brain appears younger than same-aged peers. Now, it's important to note that there are numerous caveats that need to be considered when interpreting the brain age gap. First of all, as Didac Vidal-Pinheiro has pointed out, it's unclear whether the age gap really reflects aging, or perhaps this may reflect early life or developmental effects. There's also numerous biases that need to be taken into account when interpreting the age gap. The age gap is nothing but a residual or an error term in the model, and so there are things such as regression bias, regression dilution, and regression to the mean. And the other issue is that it's often difficult to map the gap to specific phenotypes that make up the model. But notwithstanding these caveats, we can think of the brain age gap as a biological clock of sorts, right? So if the gap is greater than 0, that means the clock is running faster than normal, and if the gap is less than 0, the clock is running slower than normal. And so in this work, we've extended the concept of the brain age gap to multiple organ systems. And so as I'll show in a moment, this multi-organ characterization of biological age can be computed using data that is now routinely collected in the clinical management of older individuals. And so the broad vision here is that if we can elucidate factors that drive the divergence of an individual's chronological age from their so-called biological age, we can potentially identify strategies to slow the aging process, and also identify individuals and particular organ systems where the clock is running faster than normal, and potentially intervene before cellular deterioration accumulates and leads and manifests to disease. And in a moment, I'll show you that actually disease and these clocks are difficult that they're related to each other. So this is an overview of our study design. The study was conducted primarily using data from the UK Biobank. We considered solid organ imaging data, blood-based markers, as well as physiological measures. And each of these, we considered thousands of measures, and each measure was categorized according to 3 brain and 7 body systems. So the 3 brain systems were grey matter, white matter, and functional connectivity, and the 7 body systems are those that are shown on the right of the slide. So for example, if we look at the metabolic system, some of the features that we considered there was glucose levels, lipid concentration, and other blood-based markers. And then, just as we do for brain age, we took these organ-specific phenotypes and trained a model to predict an individual's chronological age. And we could then compute the equivalent of an age gap for each organ system. Using these age gaps enabled us to test whether the organ-specific age gaps were associated with various lifestyle and environmental factors, disease, and also to test or to use these age gaps to predict mortality. So this is just a quick overview of the key aims of the study. As I've said, we want to map the influence of an organ's biological age on other organ systems, particularly looking at the synchrony of clocks. So if one clock, for example, if the heart clock is running faster than normal, does that have an influence on our brain age, on the running of the brain clock? We also wanted to elucidate body and brain age profiles characteristic of chronic disease, identify lifestyle factors that are associated with age gaps, and also test whether we can predict mortality using the body and brain clocks. So this is an overview of the accuracy of our models. I'm not going to spend too much time on this. In the top left, I'm showing the model that incorporated all of our body phenotypes. And so the accuracy between, the correlation between predicted and chronological age was 0.79 with a mean absolute error of 3.7 years. If you look at the organ-specific models, you'll see that the accuracy is somewhat less. However, in all cases, the mean absolute error is significantly and substantially lower than what we'd expect via chance. And this is the corresponding accuracies for our brain systems. Once again, we considered a model of overall brain age, where we incorporate all brain features, grey matter, white matter, and functional connectivity, as well as tissue-specific models. We had a model for grey matter, white matter, and functional connectivity. So once we had these models established, we could then take individuals that were not used to train the model and establish a personalised multi-organ ageing profile. So in this case, I've shown 2 example subjects. We see for subject 1, if we look at the red bar, this is the cardiac clock, or the cardiac age gap is greater than 0. So in this case, the clock's running faster than normal. Whereas for subject 2, the cardiac age is younger. And so if this age gap exceeds a threshold, which could be potentially determined using normative model, what we would hope is that we can potentially intervene at an early time in this individual and either arrest this age gap or even reduce the age gap before disease sets in. The first thing that we wanted to investigate, though, was, are there interactions, or are these clocks synchronized? So, for example, does heart age influence brain age? And to address this question, we use structural equation modelling on our organ-specific age gaps, as well as an established causal structure discovery algorithm. So what I'm showing here is the network characterising influence of body age on brain age. And we see that the strongest influence on brain age is the cardiac system, as well as the pulmonary system. So to interpret this, we see, if you focus on the link from the heart to the brain, we see that each 1-year increase in cardiovascular age explains roughly a month increase in brain age. Just a reminder, we had the overall, the body and the brain here corresponds to the model that incorporates all of the organ, all of those phenotypes that are relevant to that system. We didn't look at the opposite direction, namely the influence of brain on body, and that's because in the UK Biobank, brain imaging was performed after the acquisition or after the assessment of body phenotypes, and we wanted to make sure that our inference was consistent with the flow of the chronological order of assessments. And I also wanted to point out that while these influences are not necessarily causal in a strict sense, right, there's no intervention here, they do give us, or that they do reveal directional relationships that have been established using a process of causal structure discovery. Now, you might be asking, I keep talking about aging, but how can I really determine whether the effects that we're seeing here are aging per se, or maybe these age gaps have emerged in early life or during development, right? We can't rule out that those effects. So to address that point, rather than looking at the age gap themselves, we've looked at longitudinal data, longitudinal imaging and physiological and blood-based markers, and characterised the rate of change in age gap over time. And by looking at the rate of change, we can be more confident that what we're looking at here are the effects of aging, as opposed to early life or other confounds associated with estimating age gap. So we undertook the same analyses that I've shown here, looking at influences from one organ to the other, and this is the multi-organ aging network that we were able to map, and we see that the first thing that we see here is that there are selective and specific influences from one organ system to another. For example, as I've shown here with this characteristic pathway, we see that advanced aging of the pulmonary system leads to faster aging of the cardiac system, which then in turn leads to faster aging of the renal system and the musculoskeletal system. We see that the musculoskeletal system is an in-hub of this network. And this suggests that that aging of the musculoskeletal system is a consequence, or it's a common effect of aging in multiple other organ systems, namely the immune system, pulmonary, and the heart. We weren't able to, so you might notice here that I haven't included our brain systems in this network, and that's because the overlap of subjects that we had both longitudinal brain imaging and longitudinal assessment of body phenotypes was limited. So we've analysed brain systems and body systems separately when using longitudinal data. This is the corresponding network for our brain systems, and we see that the strongest influence is from grey matter to functional connectivity. In particular, the way to interpret the coefficients here on each edge, we see that each 1-year increase in grey matter age leads to about a month per year increase in the rate of aging of FC. We don't see the converse though. Functional connectivity doesn't influence grey matter. We see this bidirectional relationship between FC and white matter. We next asked whether there are certain lifestyle and environmental factors that associate with our organ-specific age gaps. And in this diagram, to the left of this diagram, I'm showing the factors, the lifestyle and environmental factors that are associated with older appearing organ systems, and on the right are the factors that are associated with younger appearing systems. And so I'm not going to go through this in detail, but I did want to point some of the interesting associations that we find. We see, as we would expect, smoking and deprivation are associated with advanced aging across multiple organ systems, including pulmonary, musculoskeletal, as well as our whole brain model. Interestingly, we found that early menopause in females was significantly associated with advanced age across multiple systems as well. Turning to the other side of the diagram, we see that factors such as physical activity and exposure to green space are associated with younger appearing organ systems. This includes our whole body, as well as pulmonary and several other systems. But it should be noted here that these associations are relatively weak on the right side of the diagram. This is the corresponding figure for the case of brain age associations. I won't go through this in detail. But what we did next, we looked at, so all of the results that I've presented thus far are based on healthy individuals. We then went to GP records and self-report and identified individuals that were diagnosed with one of 17 degenerative brain and body diseases. And crucially, these individuals were not involved in training the model, the brain age model. We then brought back these individuals with one of these diagnoses and computed a personalised organ aging profile for each individual. And what I'm showing here is the distribution of age gaps stratified by disease category and organ. And we can see virtually across every disease category, we see evidence of advanced aging. Perhaps this is a more interpretable visualization where each icon is only showing the mean age gap. I wanted to point out a few key factors here. First of all, as expected, we see that the organ that is known to be implicated in primary disease pathology shows the most advanced aging. So as I've shown on the bottommost row there, kidney disease, CKD, is associated most with advanced aging in kidneys. Pulmonary disease, it's the lungs, and in MS, it's the brain. That's what we'd expect. But interestingly, we see that for brain-based disorders, such as as I've highlighted here, in the case of dementia and schizophrenia, we see significant and substantial aging of non-brain systems. So, for example, in dementia and schizophrenia, we see aging of the pulmonary system. And conversely, if we look at our brain age model, we see across all of these disorders evidence of substantially advanced brain age. And so this distinction between a body and a brain disorder becomes murky in the context of biological aging. And so this highlights this effect further, where I've summarized this in terms of body and brain clock. Each hand on the clock indicates the mean age gap. And so the point that I wanted to get across here once again is that for brain-based disorders, such as schizophrenia and dementia, we see significantly advanced aging of body systems. And conversely, for disorders that would be typically implicated with as a body disorder, we see significantly advanced aging of brain systems. This is an alternative representation where the size of the text is modulated by the size of the mean age gap, or in other words, how fast that organ's clock is ahead of time relative to normative age peers. We then asked whether biological organ aging is distinct from the pathological features of disease. In other words, can the effect of aging be distinguished from the effects of disease, or are physiological manifestations of disease and aging one and the same? This is a bit of a complex figure, but what I'm showing here on the y-axis is effectively feature importance in our brain prediction model. So the larger the value on the y-axis, the more important that particular feature was in predicting chronological age. On the x-axis here, we've taken those same features and just done a between-group t-test between healthy individuals and those individuals that were diagnosed with one of these 17 conditions. And we see an association between these two, and so this suggests that the more important a feature is in predicting chronological age, the greater the between-group difference between healthy individuals and those with chronic disease. And so what this suggests is that the hallmarks of biological aging, at least to a certain extent, co-vary with pathological features of aging-related disease. We had information from UK death registries on individuals who died up to 13 years following their body assessments as well as imaging. And we found that across all of the organ systems that we studied, that the organ age gap was 1 to 3 years, 1 to 3 years advanced in individuals who were deceased compared to individuals that we determined to be living 13 years up to 13 years post-assessment. So based on this, we asked, well, can we use these organ-specific age gaps to predict mortality? And indeed, as I've shown here, we trained a Cox proportional hazards regression, and we found that particularly body age gaps were potent predictors of mortality. And this is above and beyond the effects explained by age, sex, comorbidities, and other confounds, which are shown here with the blue bar. So we achieved an AUC of around 0.8. The most potent predictors were our overall body model, as well as pulmonary age. In particular, each 1 standard deviation increase from the mean for our body or pulmonary system associated with a more than 10% increase in mortality risk. So this suggests that we can use our organ-specific age profiles as in a potential model to predict mortality. We also trained models to predict survival time as well as premature death, and we see that in both of these cases, including body age gaps significantly improved our prediction accuracy. Critically though, we found that including brain age gaps did not improve prediction accuracy. So in terms of mortality, survival time, and premature death prediction, it's the body organs that are most predictive. So in summary, what I've shown here is an approach where we've generated the concept, that we extended the concept of a brain age gap to multiple organ systems, brain and body organ systems, and we've developed a personalized body and brain biological clocks. I've also shown how we can use this approach to understand synchrony between these organ-specific biological clocks, and I've characterized specific and selective influences where the aging of one organ system leads to faster aging of other organ systems. And we've characterized disease-specific organ aging profiles and demonstrated that body age gaps can be potent predictors of mortality. So this work was led by Ye Tian, this year's Carl Zales Award winner. I'd also like to acknowledge the contribution of our collaborators in this work, particularly the fantastic contribution from Michael Breakspear. So in the second part of my presentation, I'm now going to focus on a slightly different area that we're focused on. This is something that some of our more traditional work, and here I'm going to show how harnessing individual brain variability can be used to enhance brain stimulation therapy. And I'm specifically going to focus on our work in TMS. Sorry, I had a video there. I'm not sure if it's going to play. So TMS, or transcranial magnetic stimulation, is used to treat depression that has not improved with other measures. And in a nutshell, a coil is placed on the scalp, and a pulsating magnetic field induces an electric current in the underlying cortical tissue. And this is a robot that's delivering stimulation at Australia's first personalised TMS clinic. Now, the problem with TMS at the moment is, particularly in depression, is that 40 to 50 of patients show poor treatment response. And the question is why? And what can we do about that? Can we reduce this heterogeneity in treatment response? And we believe one of the key reasons for this heterogeneity in treatment response is that TMS is currently administered using a one-size-fits-all paradigm. And to elaborate on that, Current TMS protocols, when we're determining a cortical target, which is typically within the dorsolateral prefrontal cortex, we use the same stimulation target for all patients. And one approach to choosing that target is to look at a group-level functional connectivity map to the subgenual cingulate, as I've shown in this individual, and choose a site that shows maximum anti-correlation with the SGC. And then for all patients, stimulation is delivered to that precise coordinate. And we've got empirical evidence, as I've shown here, as well as some theoretical arguments for choosing such a site. So you see here, the y-axis shows improvement in symptoms, this is using a depression scale of the MADRS, and the x-axis here shows functional connectivity at the individual stimulation site. And we see here with the green individual who was targeted at a region that only shows modest resting state function anti-correlation with the SGC, treatment response was minimal. In contrast, the individual shown there in red, who was targeted exactly at that hotspot, at the point of maximum anti-correlation, treatment response was greatest. Importantly, we see extraordinary individual variation in the functional connectivity architecture of the DLPFC. So here I've shown 4 individuals, and the black circle indicates the site of maximum anti-correlation. This would be one of the this would be the candidate site for stimulation. So as you can see here, if we use a group average map to identify that coordinate, it's inevitable that for some individuals, we're going to be targeting the wrong region. In other words, we're targeting a region that doesn't show maximal anti-correlation to the SGC. And critically, as I've shown on the second row there, we can reliably identify the site of maximum anti-correlation. So this is in the second row there, I was showing a scan, an fMRI scan that was taken 7 to 10 days later, and we see consistency in the optimal site across individuals. So we've developed a methodology and a TMS protocol to personalize the stimulation site. We're now implementing this protocol clinically in Australia as part of a new TMS clinic. And the basic the basic protocol here is we first assess patient suitability. We take a 20-minute resting state functional MRI scan prior to treatment. And we've developed a novel methodology to compute personalized stimulation coordinates. Once the coordinate is established, we then use neuronavigation, as well as the robot that I showed on the earlier slide to deliver precision stimulation to that coordinate. So the innovation here is really in the methodology that we've developed to compute the optimal coordinate. One of the challenges here with fMRI is the low SNR, and the need to acquire an fMRI scan within a reasonable amount of time. And so we've developed an approach, this is work by Robin Cash, where we can now localize the optimal coordinate, in other words, the coordinate that shows maximum anti-correlation within the DLPFC to the subgenual cingulate with a precision of 2 millimeters. And we think this is well within the focality of current TMS technology. I'm not going to go into the details of the methodology here, but check out the paper. We've got a few variations of the methodology. There's a searchlight approach as well as a cluster approach. The key point here is we can reliably and accurately identify the putatively optimal coordinate. We've undertaken some evaluation of the clinical utility of optimization. This was a retrospective analysis where we went back to a TMS clinical trial that was conducted several years ago. There was no personalization in this trial, no site personalization. However, an fMRI scan was acquired prior to treatment. And so we went back to these fMRI scans and used our methodology to compute the optimal stimulation coordinate for each individual. We then computed the distance between the retrospectively computed optimal coordinate and the actual stimulation site to which treatment was delivered for that individual. And we hypothesized that the clinical improvement would be greatest in those individuals where the stimulation site was serendipitously chosen to be close to the retrospectively computed optimal site. And in fact, we found a strong relationship between improvement in symptoms, which is on the y-axis here, and the distance between the optimal and the actual stimulation site. So this gives us retrospective evidence of the benefit of site personalization, at least in the context of DLPFC, TMS in depression. And this result was recently replicated using an independent data set by an independent group of investigators. And once again, we see a relatively strong relationship between clinical improvement and the distance between the actual stimulation coordinate to which treatment was delivered and the optimal coordinate, in other words, the coordinate that shows maximum anticorrelation to the SGC. So implementation and clinical translation so far, there are several research and clinical groups that are now implementing the methodology that I've described here. Last year, the methodology was approved by our Therapeutic Goods Administration, and we were able to launch the first Australian clinic to offer personalised TMS. The clinic came online late last year, and we've already had 17 patients that have gone through the clinic, where we have used this methodology to compute the optimised coordinate and deliver stimulation to that coordinate. So the clinic was established by my collaborators, Luca Cocchi and Bjorn Berger. I'd also like to acknowledge the fantastic efforts of Robin Cash, who developed the personalisation methodology and has really led this work. So in the remaining time, I want to focus on some of our more traditional work on mapping and modelling connectomes. This is some work from Sina Manser. As most of you know, when we're mapping connectomes, this is usually done with reference to a parcellation atlas. I've shown two atlases there on the bottom, the Glasser and the Gordon atlas, and there are several limitations and issues in using an atlas to map a connectome. In work that Sina has undertaken, he's using a parcellation-free approach where we map the connectome at high resolution, at the resolution where each node corresponds to a vertex on the cortical surface. And this introduces numerous computational challenges. These networks typically comprise in the order of 100,000 nodes. And so Sina has developed various computational approaches to make the analysis of these connectomes manageable. One is a connectome spatial smoothing approach. So this enables us to smooth a connectivity matrix. So those familiar with image-based smoothing, we typically have a kernel and we apply that kernel over the extent of the image. When we're smoothing a connectivity matrix, we have a bivariate kernel. The kernel resides at both ends of the connection or at both ends of the streamline. So Sina's developed some formalisms to enable smoothing of a matrix. I think most of you guys would have used cluster-based statistics in your research, either through tools such as Randomize or AFNI or SPM. And in traditional cluster-based statistics, a voxel can be clustered if they're spatially neighbouring. We've proposed a topological cluster statistic, and in this case, we can also allow voxels that are spatially distant from each other to be clustered, to form part of a cluster, if they are anatomically connected. In other words, if there is a streamline that is found between them. And this enables us to find clusters that have anatomical support or which are consistent with the underlying structural network. So this improves the interpretation of our cluster-based findings and also leads to improved statistical power. The Melbourne Subcortex Atlas. This is work once again by Ye Tian. This is where we've developed an approach called gradientography. This is an fMRI analog of diffusion MRI tractography. And using the gradientography approach, we've mapped a hierarchical parcellation atlas of the human subcortex. And Ye has integrated the subcortical atlas into various cortex-only atlases. And so this enables us to map truly whole brain connectomes that incorporate both subcortical and cortical regions. This is work from Kaya Segwin on network modelling of brain communication. The basic idea here is that we know if two regions are anatomically connected by a fibre bundle, they can easily communicate with each other. But what happens in the case if we have a pair of regions that are not directly connected by an axonal fibre bundle? There must be some kind of polysynaptic or multi-hop communication between those regions. And Kaya has proposed various communication models, as I've shown there on the top right, to explain signal propagation through a structural brain network. And to evaluate these models, Kaya looked at the F-TRACK dataset, which is a collation of intracranial EEG recordings and more than 2 million single pulse experiments. And he's used these various communication models to predict the propagation of the signal through the structural network. And we find that Euclidean distance is one of the strongest predictors of transmission through the network. So regions that are close together are more likely to receive the signal from the source. However, there are particular communication models, as I've shown there on the right, that can explain signal propagation through the connectome above and beyond what is explained by Euclidean distance. This is some work by Marie DiBiase. We've seen this year at OHBM lots of work on the concept of brain aging and developing normative models, and normative models for brain age phenotypes. Most of these models are established using cross-sectional data, and I've shown an example of such a model here for fractional anisotropy, which is a particular white matter, a measure of white matter, and I've shown the percentiles here. And what Maria is asking here are the age-related trends that we can infer from cross-sectionally trained normative models. Are they accurate? And so if you look at the bottom plot there, what I've shown here is the rate of change, and the dashed line is the rate of change inferred from our cross-sectional model. And I've also shown a solid line there where we've estimated the rate of change from longitudinal data. And we can see here, what we can see here is that the cross-sectional estimate of the age generally underestimates the rate of change. So this points to the importance of at least testing our cross-sectional models using longitudinal data. We've also aimed to predict individual trajectories using these cross-sectional estimates, and we find that it's really hard to predict individual trajectories due to the extraordinary individual variation across the cohort. So the black lines here are our predictor trajectories, whereas the red lines are the actual trajectories that have been measured using longitudinal data. And I've only shown results here for fractional anisotropy, but we see these same effects, namely an underestimation of age-related trends across numerous brain imaging phenotypes, including volume, cortical thickness, and functional connectivity. So the key take-home message of this work is of the importance of using longitudinal data to understand the age-related trends that we're seeing in our normative models. So to wrap up, I'd like to thank the contribution of the researchers in our group to this work: Robin, Maria, Kaya, Vanessa and Sina, as well as all of our PhD students. I'd particularly like to thank Michael Breakspear and Fernando Colamanse to their incredibly important contribution to the work that I've presented here, as well as Luca Cocchi and Bjorn Berger for their efforts in establishing the Neurostimulation Centre. All this work would not have been possible without access to various open data resources, including the Human Connectome Project, UK Biobank, and the F-TRACK resource. And I'd also like to thank our funders, particularly the Rebecca Cooper Foundation. So that's all from me, and I'd be more than happy to take any questions on our research, and thank you very much.
So thank you very much for this fascinating overview on variability in brain and also body aging and the interactions. Talk is open for questions. We have some here in the app. No, there's some.
Yeah, we have a lot of questions, and thank you also for your great talk. So I'll go with the more the favorite one from the people in the app. So why is it interpreted as age gap rather than a failure to predict the actual age?
Yeah, I mean, this is a fundamental question in brain age prediction or prediction of any organ system, and it's important to remember that when we're predicting and we're considering the age gap, that this is effectively an error term. It's a residual in our model. And so, as I've pointed out, we need to be really cautious about how we interpret this age gap, and this could potentially represent early life effects. There are numerous biases in the age model. There's regression to the mean, there's regression dilution. There's also work that's shown that these models are difficult. So what we're doing here is we're really characterizing an organ system using one measure, and we might want to trace back how that single measure relates to a particular phenotype that's used to drive that model. And there's work that's shown that that's actually quite challenging to do. So this is certainly a limitation of the age gap concept.
There's a nice comment here. It's not a question, but I think it's fantastic. It's like, mate, how do I create these figures? So beautifully expressed. It wasn't just a scientifically wonderful presentation. It was also beautifully brought over. So, and it got loads and loads of likes. So it's not just, you know, the one friend.
Thanks so much for the positive feedback. I just want to say that a lot of these figures are due to our research, particularly Dr. Ye Tian, who has generated these. So I think the congratulations should go to Ye.
Right, now, back to science. So the question here, so do organ age models need adjustments for sex and race, ethnicity?
Yeah, I mean, that's a very good question. I haven't pointed out here that all of these models were stratified by sex. So we had a separate model for males and another for females. That's a really important point. Ethnicity and race are also important factors. It's quite challenging to address this using the UKB sample. It's predominantly a European, or actually a UK sample, so addressing these issues in the UK sample is difficult. We did do some supplementary analyses that I haven't presented here, where we've actually looked at organ age gaps across different ethnicities, and we don't see substantial differences across these groups. I haven't presented that data here, but yeah, it's absolutely right. This is an important aspect that we need to build into our models, and it also reflects a limitation in the cohorts that we're using to train these models. We just don't have that diversity within the cohorts to be able to model ethnicity and effects, even effects such as deprivation. Can be quite challenging to capture within the model. So just to summarise, in our models, we've trained a separate model for males and females, but in the results presented here, we haven't modelled ethnicity or race, included race in the model.
Another very popular question is related to development. So it says, a negative brain age gap could also be unhealthy, especially in developmental populations. Can you comment on the different interpretations of brain age throughout the lifespan?
Yeah, this is another important point, and it gets back to the point of interpreting age gaps. And absolutely, in a developmental cohort, a negative age gap can have a profoundly different interpretation than what it would have in an aging cohort. And this is something that needs to be considered carefully if we are going to apply these models to developmental cohort. In the results that I've presented here, these are all older individuals, 40 plus, so we didn't really have that consideration. But yeah, I agree with the questioner. We need to be cautious when interpreting the age gaps, particularly in certain cohorts, such as developmental and early life cohorts.
Well, there are many more questions, but due to time constraints, we're going to have to wrap up here. I'd like to again thank you very much for this wonderful presentation. And maybe if you'd be around and there are people that don't have to go to other sessions or whatever, they might be able to ask you your questions personally.
Absolutely, and thank you so much for chairing the session and giving me the opportunity to present some of our group's research here. Thank you.
Thank you.